Hello everybody, and thank you for joining this presentation. My name is Fatima Zohra Smaili and I am a PhD student at King Abdullah University of Science and Technology in Saudi Arabia. Today I will be presenting our work on predicting candidate COVID-19 drugs by using machine learning. The number of infections and deaths related to the COVID-19 disease are still dramatically increasing even after 9 months since the pandemic has started. So while we are waiting for a safe and working vaccine, there is also a pressing need for a drug or treatment which can actually be just as efficient in stopping the pandemic as a vaccine would be. Developing a new drug, however, is expensive and requires much more time than we can afford in the current circumstances. This is why there was a lot of experiments that have been directed towards trying to investigate the possibility of repurposing some of the existing drugs for the treatment of the COVID-19 related disease. The bottleneck here, however, is that we cannot test all of the drugs that exist in order to identify those that might be efficient in treating the COVID-19 disease, which is why in this work we try to use machine learning in order to identify those promising drugs which are worth the time and the cost of an experimental investigation. So in order to identify the potential drugs, we use here the pseudo-ontology, which is a community ontology or community-based ontology that has been created by biomedical experts as a way to unify and support the sharing of different aspects of information about coronaviruses, including the SARS-CoV-2 virus. And it basically includes information from both experiments and the literature. So our objective here is to try and leverage the information within the pseudo-ontology in order to try and predict promising candidate drugs to treat the SARS-CoV-2 related disease. In order to analyze the pseudo-ontology, we use our machine learning-based method, OPA2VEC. So OPA2VEC is a machine learning method which basically tries to generate vector representations of ontology classes and biological entities by using information from the ontology, which includes the logical axioms and the metadata, such as the labels, synonyms, etc., and by also using information within the literature as background knowledge. So this is a sort of more detailed workflow for the OPA2VEC method. So OPA2VEC combines semantic reasoning with statistical representation learning. So the way it works is that it generates two corpora. One corpus includes axioms which describe relations between biomedical classes, and also the associations between biomedical concepts or ontology classes and biological entities. And it also has another corpus which includes the metadata within the ontology, or basically the labels, the descriptions, the synonyms, etc. We then apply a neural language model on this corpus, which has already been trained or pre-trained on millions of biomedical abstracts from PubMed. This pre-trained neural language model is applied on our corpus of axioms in order to generate vector representations of both biomedical concepts within the ontology and the biological entities that they are associated with. So as a first analysis, we have applied our OPA2VEC method on pseudo in order to generate vector representations of drugs within pseudo. And then we have basically plotted them in 2D. And we have colored every drug depending on whether it has already been approved for the use on COVID-19 patients or not. So in the graph, the orange points are basically drugs which have already been approved for use against COVID-19 versus all of the other drugs which are shown in blue. So we see that there is a sort of similarity or some sort of clustering of the drugs which have already been approved for the use against SARS-CoV-2. In a second analysis, we use OPA2VEC in combination with the gene ontology in order to generate vector representations of proteins, and then we visualize them into the space again, and we color them according to whether they are targets of drugs which have already been approved for the use against the COVID-19 disease. So in this graph, the proteins which are shown in cyan are protein targets of drugs which have already been approved for use against COVID-19, and the proteins shown in magenta are basically all of the other proteins. So here again, the conclusion is that there is a sort of similarity between the vectors of the protein targets of drugs which have already been approved, and our assumption is that if we are somewhat able to identify other proteins in this area which are similar to these known protein targets, then we may be able to identify promising candidate drugs which may be efficient in the treatment of the COVID-19 disease. So to answer this question, we basically want to identify new candidate drugs for COVID-19 based on protein target similarity, and we do that by using again our OPA2VEC method. So this is the workflow that we have used, and it follows 3 steps. In the first step, we use our OPA2VEC method in combination with the gene ontology and protein GO associations in order to generate vector representations of proteins. And then we would split these protein vectors into 2 data sets, if you want, or 2 types. We have those proteins which are already targets of known and approved COVID-19 drugs, and we have other proteins, or basically other candidate protein targets. Then we would calculate the similarity between the vectors in set 1 and set 2 to try and identify those proteins which have a high similarity score to the protein targets of known COVID-19 drugs. Then in the last step, these newly identified protein targets are used in combination with the drug bank in order to identify the drugs which they are associated with and which we label as our candidate COVID-19 drugs. So in this slide, I share parts of our preliminary results. So we were able to identify many candidate drugs in this method, and here I show some of them along with the confidence score, and they have specifically highlighted those drugs which have already ongoing clinical trials. So these are the ones which are shown in red, and the fact that they already have ongoing clinical trials is somewhat of an evidence that, that our results make some sense, and they might indeed be basically worth the investigation. But specifically, we are more interested in investigating and exploring the drugs for which we have a high confidence score, but for which there is no ongoing clinical trial. So in our work, we have basically used a combination of machine learning and ontology information in order to identify candidate COVID-19 drugs based on protein target similarity. And what we want to do in the future is to explore each one of the drugs that we have identified to try and find some structural or sequential features that can give an idea about the efficiency of this drug. And we also want to explore other drugs which have completely different mechanisms of actions than the ones that we have identified. Thank you very much all for your attention, and I will be happy to take questions.